Good afternoon, and welcome to the 27th Annual Astute Clinician Lecture. I'm Jim Gilman, the Chief Executive Officer of the NIH Clinical Center. Today's special lecture is an NIH Director's Wednesday afternoon lecture series event hosted by the Clinical Center. We are pleased to welcome in-person attendees and all participants who are viewing the lecture via NIH videocast. We will begin with some general guidance. Today's CME code is 57661. Please text this code to the Johns Hopkins CME phone number on the slide to receive CME credit for this lecture. You may submit your questions for Dr. Kaplan anytime during the lecture by clicking the live feedback button located on the videocast website. And Dr. Kaplan will answer the questions as time permits at the conclusion of the lecture. The Astute Clinician Lectureship is established through a gift from the late Dr. Robert W. Miller and his wife, Haruko. I would also like to acknowledge the late Dr. John Gowan's leadership in establishing the lecture with the Millers and in navigating the partnership with FAES, the Foundation for Advanced Education in the Sciences. The Astute Clinician Lectureship highlights exciting clinical observations that have served as a focus for study. It honors a U.S. scientist who has observed an unusual clinical occurrence, and by investigating it has opened an important new avenue of research. It's now my pleasure to introduce this year's speaker, Dr. Mariana Kaplan, a rheumatologist and physician scientist. Her presentation, titled Heart of the Matter, Unraveling Cardiovascular Disease and Autoimmune Disorders, will address the scope of the problem, the putative pathogenic pathways leading to premature vascular disease and autoimmunity, and discuss potential therapeutic targets and diagnostic methods. Dr. Kaplan earned her MD from the National Autonomous University of Mexico and completed her internal medicine residency at the National Institute of Medical Sciences and Nutrition in Mexico City. She completed her rheumatology fellowship and postdoctoral training at the University of Michigan, where she was a faculty member for 15 years and an active member of their multidisciplinary lupus clinic. She is currently an NIH Distinguished Investigator, Chief of the Systemic Autoimmunity Branch, and Deputy Scientific Director for the Intramural Research Program in the National Institute of Arthritis and Musculoskeletal and Skin Diseases. Dr. Kaplan's research focuses on identifying mechanisms of immune dysregulation, organ damage, and premature vascular disease in systemic autoimmunity. Specifically, she investigates how innate immunity, in particular type 1 interferons and myeloid cells, promote autoimmune responses and end organ damage in systemic lupus erythematosus, rheumatoid arthritis, and other systemic autoimmune diseases. I promise you I'm getting near the end of this introduction. Dr. Kaplan is also interested in identifying novel therapeutic targets that may prevent premature vascular damage in systemic autoimmunity and the role of environmental triggers in the induction of autoimmunity. In addition to her research activities, Dr. Kaplan is an active clinician and teacher. She sees lupus patients at the NIH Clinical Center and is involved in developing various clinical trials for patients with autoimmune diseases at NIH. She has served in numerous roles at the American College of Rheumatology and the Rheumatology Research Foundation, the American Association of Immunologists, and the Lupus Foundation of America. Dr. Kaplan was inducted into the American Society for Clinical Investigation and the Association of American Physicians, in addition to receiving the Henry Kunkel Young Investigator Award, the Edmund J. Du Bois Memorial Lectureship, and the 2021 Distinguished Basic Translational Investigator Award, all from the American College of Rheumatology. She received the 2015 Evelyn V. Hess Award from the Lupus Foundation of America in recognition of her significant contributions to lupus research, diagnosis, and treatment. In 2016, she was awarded the Charles L. Christian Award for significant impact on the understanding of lupus. Dr. Kaplan served on the editorial board of the Journal of Immunology and Journal of Clinical Investigation and is deputy editor of Arthritis and Rheumatology. She is currently a council member of the American Academy of the Association of American Physicians. And now, Dr. Kaplan.
Thank you very much, Dr. Gilman, for this very, very kind introduction. And I feel truly honored to be giving the Astute Clinician lecture today. I want to also honor Dr. Miller and his wife and John Gallin during this lecture, and all of you for being here in person or virtually. I'm sure you have only been thinking about my talk for the last 24 hours. So these are my disclosures, and the objectives were already shown. So we know that inflammation plays or is a strong driver of the development of atherosclerosis in the general population, and this now seems like obvious, but it wasn't the case several decades ago. And one of the reasons that led to the realization that inflammation is important in cardiovascular disease is the observation that was made by many that individuals affected by chronic inflammatory conditions, particularly patients with systemic autoimmune diseases, have a significant increase in developing heart attacks, strokes, and other cardiovascular complications. And here I'm showing a non-exhaustive list of some of the autoimmune diseases that have been associated with premature vascular damage. And in red, I'm showing the ones where the epidemiology is more striking because these are more common systemic autoimmune diseases, so observations are really pointing toward an enhanced cardiovascular risk. But even in the less frequent systemic autoimmune conditions, we now know that these patients, one of the most significant causes of morbidity and mortality is premature vascular damage. And so what lessons can we learn from understanding why premature cardiovascular disease develops in patients with autoimmunity? And can these be translated also into better understanding this complication in the general population? So one question would be, are there common mediators that promote premature atherosclerosis in autoimmunity similar to what happens in the general population? So is this all high-sensitivity CRP and low-density lipoprotein? Or are the mediators that cause premature atherosclerosis really more disease-specific? Is all atherosclerosis created the same? Or perhaps the truth lies somewhere in between, that in some cases common pathways are present, but there may be distinct situations where other mechanisms may be important in promoting cardiovascular disease. And the disease that my lab has focused on for a good number of decades, and the one that I will be talking primarily during my presentation as an example and as a model to better understand cardiovascular disease development in autoimmunity, is systemic lupus erythematosus, or SLE. We now view lupus as a syndrome more than a disease, so there are many different ways by which we may get lupus, but there are some commonalities that we understand. One, that this is a disease primarily of young women at a ratio of 10:1, that because this is a syndrome and because of other factors, the condition is profoundly heterogeneous at the clinical level, and patients go throughout their lifetime with periods of flare and remission that affect them and can happen spontaneously. And we know this as the disease of the 1,000 faces, or the great imitator, because it can present in so many different ways and can affect pretty much every organ in the body. There are a few things that are also common to lupus patients that I want to emphasize because they will be important to understand some concepts during my talk. One is that one of the mechanisms that appears to be important and fairly shared among lupus patients is this dysregulation in the type 1 interferon pathways. So type 1 interferons are these important antiviral cytokines that may increase in levels during an acute or chronic viral infection, but typically remain fairly silent or with just tonic production. But in lupus patients and in other autoimmune diseases, we know that these patients have increased levels of these cytokines in circulation, and also that their cells tend to hyper-respond to type 1 interferons. And this is important because it has been proposed that dysregulation in this pathway may play crucial pathogenic roles and play a role also in organ damage. Now, one striking shared factor, and what I'll be focusing on for the rest of my talk, is that premature cardiovascular disease in lupus is an important cause of morbidity and mortality. Now, this observation is not new. Almost 40 years ago, Mary Horowitz at the University of Toronto published this bimodal peak in mortality in lupus, where early on, after diagnosis, patients were dying primarily from acute lupus manifestations and or infections secondary to immunosuppression. And then there was this second peak, much more pronounced, that happened a few decades later, many times when patients were doing fairly well, that was considered to be secondary to cardiovascular disease. And for many years, many rheumatologists thought that this was just an effect of corticosteroids and that the culprit was mostly medications and not really thinking so much about what lupus per se may be doing to the vasculature. Now, what Dr. Horowitz observed almost 40 years ago remains true and more than ever, and that vascular events are a substantial cause for mortality in lupus. And in fact, in young women with lupus, the risk for myocardial infarction is increased up to 50-fold compared with age-matched healthy women. And this is not due to vasculitis, as was originally thought, but in fact, it is from bona fide atherosclerosis. Now, this happens all over the world and is independent on socioeconomic status or ethnicity. I can show you here, for example, a recent graph from Sweden showing the percentage of cardiovascular event-free survival in the Swedish population. These are healthy individuals, and these are lupus patients. And you can see that as during a decade, women with lupus do significantly worse. This is a study from South Korea, also fairly recent, showing cumulative incidence of cardiovascular events. These are healthy people. These are lupus patients. Again, there is a big gap. And this is an interesting study that was performed here in the US that was looking at the proportion of cardiovascular mortality over the last 25 years. So in the top, you see the healthy or the general population, where, as we know, cardiovascular mortality has gone down. It has also gone down for certain chronic inflammatory diseases, including inflammatory bowel disease, rheumatoid arthritis, and psoriasis. It has actually gone up for some chronic infections, such as HIV and hepatitis C. And this is lupus, and you can see it's completely flat. So despite significant advances in treatment of lupus patients, and despite the fact that in other chronic inflammatory diseases or immune diseases we see a decrease in mortality, this is not changing in lupus. So we're clearly missing something here. Something that is important to emphasize is that traditional risk factors can only partially explain this increased cardiovascular risk. So if you adjust for tobacco, dyslipidemia, diabetes, we still see a very significant increased risk in patients with lupus. So there's something else going on. And even if we have very clear guidelines for patients with diabetes, for example, lupus patients are more likely to die following their first heart attack than patients with diabetes. And overall, the average age of the first myocardial infarction in lupus patients is about a decade earlier than in the general population. I also want to emphasize that this vasculopathy that we see as a risk factor to develop atherosclerosis does not only contribute to heart attacks and stroke, but it has been associated with progression to end-stage renal disease, to CNS involvement, to the development of pregnancy complications that are quite prevalent in lupus patients, preeclampsia, eclampsia. And it's also a concern in kids who develop lupus. These kids, even at very early stages, have significant accrual of carotid intimal medial thickness, for example. And in this case, statin trials in these kids have not proven to be effective. It is interesting also to emphasize, and this is something that we have been noticing since I got involved in trying to investigate what is going on with cardiovascular disease in lupus, that this vascular damage appears to happen very early on during the course of the disease, and sometimes may even be preceding the diagnosis of lupus. And this has been assessed in a variety of ways, where we looked at patients that have no history of prior vascular event, no traditional risk factors for the disease, and yet we find that when we look, we see evidence of vascular dysfunction, of plaque development, of myocardial perfusion abnormalities in a significant proportion of these patients. So I want to tell a little bit how I got involved in these studies and what led me to study cardiovascular disease in lupus. So I actually became interested in studying lupus when I was in medical school, when I encountered and saw a few patients that had extremely severe disease. They were teenagers, and I remember very vividly how a couple of them had developed heart attacks in their 20s and 30s. And when I moved to the University of Michigan to study, to train in rheumatology, I joined the lab of Bruce Richardson, who at the time was studying autoreactive, how the mechanisms that would lead to T-cell autoreactivity. And my project, one of my projects was focusing on how these autoreactive T cells kill antigen-presenting cells and may drive some downstream effects in autoimmunity. And I still was very curious and puzzled about this premature cardiovascular disease in lupus patients. So I started asking, well, endothelial cells can also behave like antigen-presenting cells when exposed to certain pro-inflammatory cytokines. Could some of these immune-mediated mechanisms be involved in damaging the endothelium? And I had the opportunity then to work with at first my clinical mentor and then a great collaborator, Joe McCune, and together at Michigan we established a cardiovascular lupus cohort to be able to study these patients prospectively and try to do some translational studies. And by working with a couple of cardiologists that were there at the time, Sanjay Rajagopalan and Robert Brook, they really taught me a lot about ways to measure vascular dysfunction in patients that we were able to use to establish our lupus cohort in Michigan. And then when I moved to the NIH, I really wanted to continue and expand these studies, and I got a lot of support from my bosses at NIAM, so my scientific director, John O'Shea, and clinical directors first, Richard Seal, and then Bob Colbert, really supported me continuing with these investigations. And by working with Sarfaraz Hasni, by revitalizing the lupus program and establishing a lupus cardiovascular cohort here at the NIH Clinical Center, and then working with Nehal Mehta, which at the time was at NHLBI, we really were able to kind of continue the studies I had started in Michigan, but expand them to do what we wanted to do, more impactful work. And so what is unusual about the development of atherosclerosis in lupus, and what hints is this giving us regarding what may be happening? So for one thing is this is a disease of young women. Young women should not be getting heart attacks and stroke. What we call the classical inflammatory burden that has been proposed by Paul Ridker and others as being a main driver of atherosclerosis in the general population, high sensitivity CRP, high low-density lipoprotein, really does not apply to lupus. Most lupus patients don't go around having high levels of C-reactive protein, and there's a physiologic explanation for that. And in fact, when we see elevations of CRP, we usually suspect infections. And as I'll mention later, we have found that low-density lipoprotein does not appear to be the key lipoprotein mediating atherosclerosis in lupus patients. And the last thing I want to mention is that both in adult and a pediatric population, although small, the statin trials have been pretty inconclusive. So we really don't know how effective statins are in compared to what we see in the general population in patients with autoimmunity. So I'll tell you a little bit about this cohort we have established at the NIH Clinical Center. These are patients that come to the clinic, have very deep clinical phenotypic assessments, and then we obtain blood, we do a number of experiments, we biobanked, we do assessments of vascular function in different vascular territories. We do imaging of the aortic wall to assess vascular inflammation using FDG PET-CT. And we also perform CT angiograms to assess the quality and the quantity of the coronary plaque. And by doing that, we have been able to continue, and with something that is ongoing, trying to understand the natural history of cardiovascular disease development in lupus, but also use this as a way to compare the vascular natural history of lupus to other chronic inflammatory conditions. When possible, see what happens when very distinct insults happen that may affect the vasculature, for example, certain microbes, and then to enroll patients in investigator-initiated clinical trials that have as either primary or secondary outcomes vascular disease or vascular dysfunction. So just to show you how dramatic this is an observation we made 20 years ago when I was still at Michigan, where we took very young lupus patients that had recent diagnosis of lupus, and we performed vascular studies to assess endothelium-dependent responses and vascular smooth muscle responses, and compared their vascular endothelial function and vascular smooth muscle function to healthy people and to a much older cohort of patients with well-established coronary artery disease and many risk factors. And what was striking at the time is that even these young lupus patients with recent diagnosis of lupus had the vascular health of these much older patients with a lot of other risk factors, whereas their vascular muscle responses, at least at that time, were fairly well preserved. So this suggested to us that something quite dramatic was happening at the level of the endothelium early on during the course of the disease. And more or less at the same time, we were having other groups reporting, for example, this is a paper by Jane Sammons' group at Hospital for Special Surgery that showed that when you look at people in their 20s and 30s and you compare lupus patients to healthy controls, you can see that they have already, almost 15% of them already have pretty significant carotid plaque development. And of course, getting older and having lupus is not a good combination. And by the time they are on their 6th decade of life, about 75% of patients already have a lot of carotid plaque compared with 30% of controls. And as we have continued looking at these studies, looking in different populations, different ethnic groups, and with new techniques, we can see that this has remained the case 20 years later. This is a study done a few years ago where we showed here that when we look at vascular stiffness through this technique called cardioankle vascular index, there's a significant increase in arterial stiffness in lupus patients compared to controls, again suggesting premature arterial aging. And just to show you a couple of illustrative pictures from the studies we're doing here at the clinical center, this is an FDG PET-CT scan that looks at aortic wall inflammation, which has shown to be not only a surrogate of inflammation, but also a predictor of potential future development of atherosclerosis. And, you can see very clearly this is a young, healthy woman, and this is a young lupus patient, how much inflammation you can detect in the vascular wall. And this associates with the fact that these patients, even at a very young age, when we look at their coronary arteries, you can see these bulges here where the arrows are that represent non-calcified coronary plaque burden. So overall, what we appear to be seeing is that not only they have more coronary plaque, but the quality of this plaque appears to be different and appears to be some sort of more unstable plaque that may be more likely to rupture and cause acute coronary events. Sorry, I think this is stuck. Oh, sorry. So what drives premature vascular damage in lupus? And this is complex as lupus is, because again, as I mentioned, this is a syndrome, and there are many, many factors that become dysregulated in lupus. Some are more lupus specific, because any immune cell type you look at some point will be abnormal in lupus patients, and there are also a number of humoral factors, autoantibodies, complement consumption, etc., that hypothetically could damage the vasculature. There are also many non-lupus specific but highly prevalent factors, cardiometabolic factors that are abnormal in lupus patients, insulin resistance, renal disease, and even genetic polymorphisms that may make these patients more predisposed to vascular damage. So how do we dissect this? So one of the initial observations we made many years ago was that as we were studying the hypothesis that endothelial cells become damaged by certain components of the immune system, one idea we had is that these cells were being killed and accelerated pace through immune-mediated insult. So we developed this assay where we looked at the levels of circulating apoptotic endothelial cells, thinking that these cells were being damaged and were being released to the circulation. And in fact, we found that lupus patients had tons of these apoptotic endothelial cells circulating, whereas patients with well-established coronary artery disease but no lupus didn't show that. And then when the endothelium becomes damaged and endothelial cells die, under normal conditions, there should be proper vascular repair mechanisms that will re-endothelize the vasculature and promote repair. And typically, this has been described to happen through endothelial progenitor cells and other cells that have the ability to get incorporated into the damaged blood vessels and revascularize. But what we found in lupus patients is that there was this imbalance between endothelial cell damage and vascular repair, where, for example, the number of endothelial progenitor cells in lupus patients was significantly lower than what we would see in healthy controls, despite the fact that this should have made the bone marrow really respond with higher levels of these cells. And not only that, but those endothelial progenitor cells purified from lupus patients just does not have the ability to differentiate into mature endothelial cells. And this was the first time we really started thinking about lupus as being a disease of impaired angiogenesis. But this has remained to be the case as we have continued to study lupus patients throughout the years. So what is causing this imbalance between vascular damage and vascular repair? And what I would say as I move on is that we have really focused on what role may dysregulated innate immune system could play in this phenomenon. We think overall that innate immune system is very important in lupus pathogenesis from work done in my lab, but particularly focusing on vascular damage, we think that that is also the case. And so when we started thinking what are the potential mediators that cause this imbalance between vascular damage and repair, we tested many potential pathways. And long story short, what we found after many years of work was that the same type of interferons that I mentioned to you are dysregulated in lupus and cause changes in immune cells were directly involved in causing damage to the vasculature. And they did that in part because we found type 1 interferons significantly contribute to an anti-angiogenic signature in the lupus milieu. And they do that in part because they would directly induce apoptosis of these endothelial progenitor cells, but also because by signaling in various cells, they suppress pro-angiogenic molecules such as vascular endothelial growth factor, and then promote the synthesis of anti-angiogenic molecules, for example, IL-1 receptor antagonists. And while IL-1 may be a bad cytokine for certain inflammatory diseases, it is interesting that in the case of lupus, this suppression of the IL-1 pathway appeared to play some role in mediating abnormal vascular repair in lupus patients. And this is just the initial assessment, but then we found that type 1 interferons really play very strong pleiotropic roles in driving vascular damage in lupus. Another thing that has been reported is that type 1 interferons, for example, can promote macrophages to become foam cells, which are the very important cells that accumulate in arterial plaque. And also that type 1 interferons are stroke promoters of thrombosis. They make megakaryocytes, and then platelets that develop from megakaryocytes have a very activated phenotype that is highly conducive to clot formation. And so the studies have evolved since our initial observations to even observe that type 1 interferons can by themselves induce insulin resistance because they activate certain immune cells in the liver and promote this complication. Now, the other component of the innate immune system that we think is significantly involved in this premature vascular damage is this regulation in neutrophil biology. And part of this comes from one antimicrobial function that under normal conditions is important to save lives, but that becomes significantly dysregulated in lupus patients and patients with other autoimmune diseases. And this is an antimicrobial strategy called the formation of neutrophil extracellular traps, or NETs. This is a meshwork of chromatin bound to granule proteins that are released from neutrophil granules, including very potent enzymes such as myeloperoxidase and others. And this is extruded into the extracellular space. Typically, it has the ability to trap microbes and kill them. But in lupus patients and in patients with other autoimmune diseases, these nets are formed continuously, even in the absence of infection. And we have identified another imbalance, which is that lupus patients and patients with other autoimmune diseases form a lot of nets, and these nets are not being properly cleared in a significant proportion of these patients. So these structures have an extended half-life in circulation and in tissues. And we have been very focused in trying to understand what happens when these structures are formed and not properly cleared. And what we know is not only that this may play a very important role in other aspects of lupus I will not have time to discuss today, but that there is a subset of cells within lupus patients called low-density granulocytes that we identified about 15 years ago that are present in high numbers in lupus patients, and that one of the features, we do think these cells have pathogenic effects, are pro-inflammatory, and part of the reason why they are bad players in lupus is through their damage to the vasculature. So what we have found is that these cells particularly are exquisitely primed to form these nets in the absence of any obvious infection, and that these nets do a very good job at harming endothelial cells. We also have found that interestingly, and we are not sure yet why, these cells have different biomechanical properties that makes them become quite stiff. And so when they're traveling in blood vessels, they become trapped, especially in smaller blood vessels, and that causes tissue hypoperfusion and may potentially contribute to thrombosis and other vascular complications. We also have observed over the years that patients with a number of other autoimmune diseases and also monogenic or inflammatory syndromes that we have had the opportunity to see here at the NIH through various collaborators, they all have these low-density granulocytes, and these low-density granulocytes share this ability to form these nets. And what all these diseases, one of the things they have in common is that they're all associated with either enhanced cardiovascular risk from atherosclerosis or just to very profound vascular damage. So we are hypothesizing, and it's a hypothesis we're running with right now, that the presence of these dysregulated neutrophils may be a common factor that promotes premature vascular damage or enhances vascular risk. How do NETs induce vascular damage in lupus and in other diseases? We have spent many years studying this, and just to summarize a lot of work, I can tell you that the NETs from patients with autoimmunity are different than the NETs generated in infections, and they have a higher ability to kill endothelial cells. We think this is in part driven by certain matrix metalloproteinases that these NETs extrude and have an ability to induce an endothelial cell death pathway. NETs, as I mentioned, are potent inducers of thrombosis. They do a really good job at activating platelets and activating anticoagulants and inducing coagulation. And this is also happening in homeostasis, but it's exacerbated in cases of inflammatory diseases. NETs themselves may contribute to creating a vicious inflammatory cycle because while type I interference can potentially promote NET formation, NETs can also play an important role in inducing type I interference and also inflammasome activation. And these two pathways can really exacerbate vascular injury and therefore promote a vicious cycle that is hard to stop in patients with autoimmunity. I will tell you a little bit more about the role that NETs may directly play in modifying lipoproteins in a few minutes. And so overall, through preclinical work in animal models and clinical work or translational work we are currently performing, we think that NETs are playing a very important prothromogenic role in the general population, as described by our group and others, but particularly in patients with autoimmunity. And we have, in fact, found, at least in preclinical models, that inhibiting NET formation can mitigate atherosclerosis. So, this being an astute clinician lecture, I will try to make it as translational as possible and tell you a little bit how are we trying to prove that this is, in fact, relevant in the clinic. So, one of the things we have noticed is in this cardiovascular lupus cohort we have established here at the NIH, that when every time the patients come, we do a variety of measures to try to assess determinants of vascular dysfunction and vascular risk. And what we have found is that after adjusting for multiple confounders associated to vascular risk, the levels of these low-density granulocytes in circulation significantly associates with the degree of non-calcified coronary plaque burden in these patients. And furthermore, when we have done RNA sequencing analysis, assessing whether the presence of this low-density granulocyte gene signature will associate with vascular abnormalities, we have seen that having this low-density granulocyte gene signature strongly associates with having increased non-calcified plaque burden and increased vascular inflammation when compared with those patients that do not develop this low-density granulocyte gene signature. So, of course, this is not proof, but it suggests that perhaps this level of these cells, or characterizing, identifying these cells at increased levels in lupus patients, could potentially serve as a biomarker of cardiovascular risk, and ongoing studies, longitudinal studies, will hopefully allow us to address that. So overall, the hypothesis is that there is a pathophysiological alliance between dysregulated neutrophil function and type I interference that may play an important role in the development of atherosclerosis. And I think lupus really represents the extreme phenotype here, where both these pathways are so significantly dysregulated that they converge at the level of the artery and amplify pro-inflammatory signals in an exacerbated manner. But perhaps this is also relevant in not only patients with other autoimmune diseases, but in subsets of the general population. There are these observations that exposure to certain infections may exacerbate atherosclerotic risk. It's certainly been described for patients with hepatitis C, as I mentioned, HIV, and other viral infections. How might this help us in the future to further understand how these complications develop in patients that have to suffer from chronic infections throughout their lifetime? And just another piece I want to emphasize when we're starting to think about how to modulate this is that another observation we have made over the years is that the metabolism of the innate immune cells in lupus patients is dysregulated, and that many of these cells, neutrophils and other myeloid cells, have evidence of mitochondrial dysfunction. And we have actually found that both in vivo and in vitro, modulating immunometabolism in lupus models or in lupus cells leads not only to improvement in immune dysregulation, but we have hints that it may impact vascular damage in lupus. And this is very preliminary, but I want to tell you a very recent observation we have made, which focuses with a very fundamental pathway of immunometabolism that is an anti-inflammatory pathway. And this is the so-called itaconate pathway. Itaconate is a compound that is made by our bodies that serves as an anti-inflammatory and antimicrobial strategy. It's mostly made by macrophages during inflammation to protect against damage that our bodies may elicit from inflammatory responses, and it actually also regulates type 1 interferons. And in recent studies, we found that this pathway is actually important in lupus, and that actually supplementing this pathway in animal models of lupus makes lupus better. But what's interesting to observe in a cohort of lupus patients is that levels of itaconate overall in lupus are significantly decreased. And this, as an observation, may not tell us a whole lot yet, but when we do associations with vascular variables, not only lower itaconate associates with worse lupus disease activity, but those patients with lower itaconate have more coronary plaque, more aortic inflammation, more insulin resistance, and conversely, the higher the levels of itaconate, the better the high-density lipoprotein or good cholesterol functions. So this is preliminary, but we're very interested in pursuing this further to see if there are ways where we can modulate immunometabolism as a vasculoprotective strategy. Just a few words about dyslipidemia in lupus. So this has been described for many years. Many different abnormalities in lipoproteins are present in lupus, but which one is relevant in the context of cardiovascular disease? So what we have observed is that the main abnormality that we consistently see in lupus patients is not so much related to low-density lipoprotein, but more to high-density lipoprotein, or the so-called good cholesterol. And as you know, high-density lipoprotein has a number of atheroprotective functions, and it's also highly anti-inflammatory. And one of the functions of high-density lipoprotein is the ability it has to accept cholesterol from macrophages and prevent foam cell formation. And so studies done in the past have shown that the capacity of HDL to do cholesterol efflux is actually negatively associated with atherosclerotic risk. So how HDL functions is important. We found some years ago that this capacity of HDL to take cholesterol out of myeloid cells is significantly impaired in lupus patients. And furthermore, that by itself, impaired cholesterol efflux capacity is also significantly associated with this non-calcified plaque burden development. So how is this happening? And what we found is that, in fact, going back to the neutrophils, these nets that lupus patients are generating are oxidating high-density lipoprotein in very region-specific manner. And by doing that, they're altering their function. And this oxidized HDL really loses its atheroprotective capabilities. And one thing that happens is that HDL is actually a potent anti-inflammatory molecule that, when cells are exposed to danger signals, for example, toll-like receptor agonists, HDL actually activates transcription factors that lead to suppress the effect of these pro-inflammatory signals. But when the lupus HDL becomes oxidized, we have found it is incapable of doing that. And so danger signals can go through the cell and promote inflammation, and HDL really has lost this scavenging anti-inflammatory activity. So how do we put it all together? We think that these nets generated by lupus patients produce a number of pro-inflammatory molecules, oxidative enzymes, that oxidize the HDL. This impairs cholesterol efflux capacity, promoting foam cell formation. And on the other hand, because the HDL is not able to dampen inflammation, these macrophages may be induced into being pro-inflammatory, synthesize pro-inflammatory cytokines, and again contribute to premature vascular disease. So how can we target this? And as I mentioned, this is an area of great need in lupus. There is very little we know about prevention, and we know a lot about how to prevent cardiovascular disease in diabetes. The guidelines for cardiovascular prevention in autoimmunity remain very nebulous. We are told to do yearly monitoring of traditional cardiovascular risk factors, but we have no idea how useful that is. And although we think lupus should be considered a cardiovascular disease equivalent, we have no idea, again, what's the role of statins here, how do we follow recommendations for lipid management. And although there are many scores that have been developed, some even lupus-specific, to try to assess the 10-year cardiovascular disease risk score, the organization, for example, the European League Against Rheumatism even has said the actual risk is probably underestimated in patients with lupus. So this is a big problem, because until we don't really have good guidelines on how to prevent and identify patients at risk, we cannot do a very good job. There are a number of primary cardiovascular disease prevention strategies that have been proposed, and some are mentioned here, from the Mediterranean diet to using a number of immunosuppressive drugs that may potentially have vasculoprotective roles. And of course, corticosteroids are dual-edged swords, because while they have a number of negative cardiometabolic effects, we also know that they are highly anti-inflammatory, and so how much to use for how long remains a matter of debate. But all of these may be important observations, but as I showed you, that line remains flat regarding decreasing cardiovascular mortality. So clearly, what we're doing is not enough. Can we explore other strategies? So in the last few minutes of my talk, I'll tell you a little bit about what we have been doing to try to test our hypothesis. And the two main targets we have right now is type 1 interferons, that, as I mentioned, we think plays very important pleiotropic roles throughout the development of plaque and cardiovascular events, and also can we target some of these neutrophils and their dysregulation in ways that we may break this vicious cycle. So to assess the first target, which is type 1 interferons, we have followed so far two independent approaches. One is to try to see if we can block signaling of type 1 interferons through their receptor, type 1 interferon receptor. The other is whether we can go more downstream and block the signaling pathways that, by which type 1 interferons signal. And so the first approach we are following, which not chronologically the first, but the first I'll talk about, is using this drug called anifrolumab. This is a monoclonal antibody that is directed against type 1 interferon receptors and was approved by the FDA a few years ago to treat moderate to severe lupus. So following our hypothesis, we first approached the company that had performed these trials and asked whether we could get access to some of their serum and plasma samples, because the trials had been already completed. And as I mentioned before, lupus patients have these circulating nets that are really increased compared to what we have in healthy individuals, and we think nets are important in vascular damage. So we asked, can we check whether blocking type 1 interferon receptor would lead to decreases in net formation? And that was indeed the case, that patients that were receiving anifrolumab decreased their levels of nets, those on placebo went up. But furthermore, we found that when we measure the function of HDL after blocking type 1 interferon receptor with the antibody, those that had received the drug had almost a 20% increase in the function of their HDL, which is not trivial because not that many drugs can do that, whereas those on placebo, if any, they had some form of an increase, but it was not significant. So this gave us some increased enthusiasm to try to test in vivo whether giving this drug to lupus patients will lead to improvements in cardiovascular disease. So this study started this year, so I don't have results to show you, but I want to emphasize what we're doing. And we are now performing a phase 2, a randomized placebo-controlled single-center trial testing anifrolumab versus placebo. The primary goal is a vascular role. We want to look at vascular inflammation and vascular function over an 8-month period in patients who receive the drug. And as a secondary role, I think we'll learn a lot about how are we modifying cardiometabolic parameters and immune dysregulation, as well as some markers of safety and tolerability. So I'm hoping in the next couple of years we'll be able to answer whether this strategy does something to the vasculature. The other pathway that I mentioned is also relevant to have type 1 interference work is the JAK-STAT signaling pathway. So as you know, this is a fundamental pathway by which cytokines, including type 1 interferons, signal through cells. And neutrophils also have a very active JAK-STAT pathway whereby many cytokines crucial to neutrophil development and function use this pathway to exert their effects on neutrophils. And we recently published the results of a small proof of concept study where we gave patients with lupus one of the JAK inhibitors called tofacitinib for a short period of time, 2 months, and compare these results to placebo, patients that took placebo. And what was striking is that even during a very short period of time, the drug appeared to modulate this regulated innate immunity in lupus patients that we know is conducive to vascular damage. So just to emphasize some things, these low-density granulocytes went down with the drug, not with placebo. We saw decreases in net formation when the patients were on the drug. Not surprisingly, type 1 interferon gene signature also decreased. But all of this translated into finding that, in fact, patients who took the drug had significant decreases in vascular stiffness even after a short period being on the drug. And what was striking was that levels of high-density lipoprotein or high-density lipoprotein function really increased when the patients took tofacitinib when compared with those that took the placebo. So this is early on, and of course there are some things to consider regarding vascular risk with JAK inhibitors, but this is, perhaps helping us continue with this hypothesis that targeting these pathways may actually do something to the vasculature. In collaboration with Michael Sack and HLBI, who really has spearheaded beautiful studies on the role of fasting and fasting mimetics in aging and other health parameters in the general population and other diseases, we partnered a few years ago to look at the effect of fasting mimetics using NAD boosting as an anti-inflammatory mechanism. And in initial studies published a few years ago, led by Michael Sack, we found that actually NAD boosting through this supplement called nicotinamide riboside led to significant decreases in type 1 interferon responses, both in healthy individuals and in lupus cells. So Michael Sack is now the PI in this study with whom we are collaborating to try to assess the role of this immunometabolism boosting or NAD boosting using NR in lupus patients. And not only are we going to assess disease activity, but we're going to be looking at immune profiling and, importantly, at vascular function. So this could be really exciting because this is not an immunosuppressive strategy, and yet it could have some potential, at least adjuvant roles, in cardiovascular prevention in lupus patients. And finally, the last trial I will tell you about came from observing that one of the pathways that type 1 interference was significantly suppressing downstream in the endothelium was this peroxisome proliferator activator receptor, or PPAR pathway. And so with type 1 interference, when we treat cells with type 1 interference, PPAR is downregulated. And as you know, PPAR alpha, PPAR gamma, these pathways are very relevant for a number of cardiometabolic parameters in the general population. PPAR gamma agonists are used for insulin resistance and diabetes, and in many different patient populations, they have been found to have potential effects on cardiovascular risk and cardiovascular prevention. And so a few years ago, we completed a study where we treated getting stuck again. Where we completed a study where we gave lupus patients pioglitazone. This was a double-blind placebo-controlled crossover study where the patients took either placebo or drug for 3 months, did a washout of 2 months, and then cross over to the other arm. And we tried to assess whether the drug would be useful for vascular risk. And what I can show you here is when the patients were on the drug, vascular stiffness were significantly down. Then when we take them off the drug, the effects are reversed. This is placebo. When we put them on the drug, you see how vascular stiffness goes down. And this, of course, was also associated with significant improvements in insulin resistance and lipoprotein profiles, suggesting, again, another mechanism and another potential therapeutic target that was really driven by some of the observations we made in the lab and that really, give us a lot of enthusiasm and inspire us to continue these studies. So just to summarize, I hope I convey that increased cardiovascular risk in autoimmunity is a big problem. It's related to a combination of traditional and non-traditional risk factors, and it's important to emphasize that suppression of this immune dysregulation appears to be key to lessen cardiovascular risk, and that although strategies to reduce traditional risk factors should be implemented, we really need better disease-specific guidelines to better diagnose and treat these patients. And we're hoping that with some of these discoveries we're making regarding the role of the innate immune system and other pathways, we may be able to come up with more targeted therapeutic strategies that at the end what we want really is a personalized approach strategy. This is a complex condition, and until we really don't understand who is at risk and how can they better be treated, we won't fulfill our goal. And finally, I really don't have space to thank all the many people that have been involved in these studies. Here are different iterations of my lab and of the Lupus Clinical Trials Unit. Here I'm only mentioning the people in my lab who contributed directly, previous or current members who contributed directly to the experiments I presented today, but there are many more I need to thank. The Lupus Clinical Trial Units, as led by Sarfaraz Hasni, everyone involved there had been instrumental. All my support from NIAMS leadership and NIAMS collaborators, great collaborations with NHLBI and other institutes, and my collaborators at the University of Michigan and other institutions. And finally, if they let me, I was going to just thank the funding agencies, but I think I'm NIAMS and other institutions. Thank you very much for your attention.
Please, if anyone has questions, please use the mics.
Hi. Have you looked at changes in proteasomes as a possible mechanism for the inducing autoimmunity?
Regarding autoimmunity in general or the vascular?
Well, in the SLE, if you have changes in the proteasomes, then the proteins get chopped up differently and presented by MHCs differently.
Yes, so there have been some studies done, a lot pertaining, for example, to B-cell biology and plasma cell biology, where actually proteasome inhibitors are now being tested. We did years ago with a collaborator some studies looking at proteasome degradation and generation of autoantigens through that mechanism, and there indeed appears to be very distinct findings compared to healthy individuals or individuals with other autoimmune diseases. I think this is work in progress. A lot needs to be understood, but certainly there seems to be some disruptions in how the proteasome works in lupus patients compared to healthy individuals. Thank you.
Hi, Mariana. Great talk.
Thank you.
So I was wondering whether you could talk a little bit more about the molecular hierarchies between NETosis, interferon, dyslipidemia, and metabolism. Who is regulating whom?
Yes, it's a chicken or egg question, and that's the problem with studying lupus, that by the time we see these patients, everything is dysregulated. I think you can tie them together in that part of this is genetically driven, and we know that people at risk for lupus already are making type 1 interferons at elevated levels or hyper-responding. And in fact, there are also patients, before they develop lupus, they're making more NETs. So there is some crosstalk that happens there early on that leads me to believe that that's why maybe the vascular damage, by the time we diagnose the patient, they have already perhaps endured some degree of vascular disease even before they were diagnosed with lupus, and that's the problem. And I think the rest happens downstream, that you have this highly oxidative environment that ends up oxidizing lipoproteins and sort of, synergizing to cause, and that's why the vascular damage is so extreme, because you have different players, but also because the damage started probably fairly early on. Now, what started first, I don't think we can really tell it.
So, for instance, if you use JAK inhibitors, right?
Mm-hmm.
Do you block NETosis?
Yes,
You do.
you do. Yes, so we target both. So that's the beauty, I think, of that pathway, that you may be able to target both components.
Okay. Thank you.
Thank you.
Okay.
Hi, thank you for your talk. I was just wondering which blood vessels become stiff, if you could elaborate on that.
Yeah, so the tests that we use are focusing mostly on medium blood vessels, so mostly from the extremities. But there are also other studies that have shown that even so at the level of the aorta or larger vessels, you can see that. And there is also significant microvascular dysfunction. So really in lupus, all vessels are affected. For the purposes of atherosclerosis, we focus more on medium and large arteries, but the role of damage to the small blood vessels cannot be, overemphasized, because it may trigger renal disease, pregnancy complications, and other abnormalities.
Thank you.
Thank you, Dr. Kaplan. Colchicine works as a neutrophil tranquilizer. So based on COLCA trial, do you consider using colchicine in lupus patients? Do you think it works?
Yeah, so colchicine can have some mild effects on the neutrophil dysregulation. I don't think they are as effective to dampen the responses as other drugs. And of course, colchicine has some potential for chronic use in lupus patients, can have some potential issues regarding, bone marrow cellularity, kidney damage, etc., that you have to be cautious about. But certainly it's a drug that can have some mild effects on the phenotype.
Okay, thank you.
Comment, perhaps. You did not mention the calcification and platelet thrombosis, which is different than immune arteriosclerosis.
The calcification?
Yeah, you did not mention calcification and platelet thrombus.
Yeah, so it's interesting that, as I discussed, that the type of plaque that we see in lupus patients tends to be less calcified than the one described in other patient populations. And this is something that we continue to see. We think it's probably a bad thing because the plaque may be more unstable. There is some discussion about that. And platelets in lupus overall are activated through these processes, both by type 1 interference and by the neutrophil dysregulation. Certainly, platelet activation has been well described in lupus, and we think that, of course, plays an important role in acute coronary syndromes and other complications.
Okay, and this question came in a little earlier before you got into detail. Is there any reason why statins would not be beneficial? Is there a different biological mechanism or action?
Yeah, and I want to clarify. I should not say they're not beneficial. I think we actually tend to put our lupus patients on statins very often. But whether they are as effective as in other patient populations remain to be seen. The trials have been fairly small, but none of them have been really suggesting that statins by themselves are enough. So that's why we need better guidelines and larger studies to assess what other combinations are needed, what's the right dose, what type of statin may be better. I do not think we can say they don't work. There is something physiologic that make them, or pathologic that make them not work in autoimmunity, but it seems to be a different mechanism.
Mm-hmm. Do you have any closing? Thank you.
Yeah, so thank you all for attending, and thanks Dr. Kaplan for a great astute clinician lecture. And we have a reception in the medical library, especially right outside. It's not in the medical library. Don't go to the medical library. It's right outside, and especially for the young investigators, students here who might want to talk to Dr. Kaplan about the path that you want to follow to be up here one of these days. Thanks.